So first I will tell you a secret that I'm not fluent in DNA, but I can tell you some things about genome editing. So you might have read about the news about the first pig kidney that was transplanted into a man of age 62 2 months ago. What is interesting about this pig kidney is that it was genetically edited to make it more compatible with humans and reduce the chances of infection after a transplant. In other words, scientists edited the genome of the pig kidney cells to fit humans. While promising, we will not know the long-term effect of this transplant, as unfortunately this person died this week, according to his doctor, from complication unrelated to the transplant. Another field where genome editing is growing interest is agriculture, mainly due to climate change. Genome editing technology could help crops withstand the stress of climate change. For example, can we edit the genes of some plants to make them resistant to extreme weather conditions like extreme heat or cold? So now, after I presented you two applications of genome editing in agriculture and medicine, I would like to ask you a question. Can you think of some of the differences between genome editing in agriculture and medicine? So leaving aside the ethical issues with genome editing in medicine, extreme safety measures must be taken before any genome editing is allowed in this field. One of the issues, along with the unknown long effect that I mentioned before, is the issue of ensuring accurate genome editing. In other words, ensuring that only the desired modification, the desired edit, is performed, and preventing any other modification that can cause harmful mutation. The standard definition for such undesired edits is called off-targets. So in my research, I'm using AI and computational tools to help biologists analyze their experimental data. One of the questions that I'm trying to answer is whether we can utilize AI to design a safer genome editing that avoid those off-targets, those undesired edits. But before I can tell you how I'm planning to do this and what is the exact problem that I'm aiming to solve, we need to understand the technology behind genome editing, which is the common thread between genome editing in agriculture, medicine, and other fields as well. So this revolutionary technology is known as CRISPR. This cutting-edge technology derived from the immune system of ancient bacteria is the key for efficient genome editing, paving the way also for promising application in various fields. But how does it work? So scientists first identify a sequence in the DNA that they are willing to edit, a target location. For example, a location that causes health problems. Then they design a specific CRISPR system to edit that particular location. When you try to edit the DNA, the first thing that you have to do is to damage the DNA. This is done by the CRISPR system itself. It has two parts. The first one, targeting the location, and the second one, damaging it. Then after the damage is done, cellular repair processors are kicking in, and you convince those repair processes to make the edit that you want, and not the natural repair that is performed otherwise. We can also extend this analogy of CRISPR system to guided missile even further. You can think of, roughly speaking, a guided missile is comprised of two components: a warhead, which is causing the damage, and some targeting mechanism that ensures that the missile is heading to the correct location. Similarly, CRISPR is a two-part system. You have something called Cas9 enzyme, which is a protein, and something else called guide RNA. And together they form the CRISPR system. The Cas9 is kind of like a warhead. It wants to damage the DNA. But the guide RNA, it's just like a GPS coordinate. It prevents the Cas9 from damaging the DNA until it finds the exact spot where it matches in the genome. The modular part in this system is the guide RNA, which changes according to the specific location that we are willing to edit. The guide RNA is just a sequence of letters, A's, C's, G's, and T's, that matches the specific location in the genome. It's like a GPS coordinate, as I told you before, but just with letters. The potential problem is that when we are willing to edit the genome, there might be other similar sites in the genome that are similar to the guide RNA that we designed. Therefore, there might be other differences from the perfect match that we are willing to edit, and those can be potential sites to be off targets. Like when comparing words, there might be certain different types of differences between the guide RNA and the perfect match. For example, a mismatch, which is different letter in certain position, a missing letter, which is defined as deletion, or an extra letter, which is defined as insertion. And it can be also any combination of those differences. And the number of differences is usually defined as the added distance between the guide RNA and the perfect match. So the potential problem is that we are willing to edit some location in the genome, but there might be other sites in the genome that are similar to the guide RNA that we designed, and they can be mistakenly edited. So now I can redefine the abstract question that I presented to you before to the question of whether we can predict whether a guide RNA is safe to use and can avoid those off-target, those undesired off-targets. To do this, we build an AI-based model which predicts whether a guide RNA is safe to use or not. So the foundation of every AI model is data. So the first thing that we have to do is to collect suitable data to training this model. So since we want to know which are those suspect sites that are similar to the guide RNA that are prone to be off-target, we apply an algorithm which finds all the potential off-target sites based only on the similarity between the guide RNA and some genomes and the genome. After that, we have a list of suspects of off-targets that prone to be off-targets. Then, since we want to know out of those suspects which of those were really off-target or not, we utilize experimental tools, laboratory experiments that designed to find all the off-targets for those some guide RNAs. So we define those experiments found from the experiments as active off-target sites, as they were really found to be off-target. And all the remaining sites out of the suspects, we define them as inactive off-target sites, as they were found to be non-off-target at all. I'm skipping a lot of processing that is done to make the data suitable to training a neural network, an AI model. But once we have the labeled data, the only work that is remain to do is to select a suitable neural network that can learn the patterns between those active off-target sites and inactive off-target site. And actually, this model learned the biological patterns and distinguish those sites, those off-targets, from the potential. Ultimately, we would like to use our new developed AI-based model for new guide RNAs that were not examined in the experiment, and for their potential off-targets to understand their probability of being really off-target or not. Therefore, aggregating all those probabilities and knowledge will give us some indication whether the guide RNA is safe to use or not. So while it is just one piece of the puzzle, using an AI-based model to design the CRISPR system is one step in making genome editing safer. This will allow new genome editing application in various fields like medicine and other sensitive ones. Thank you very much.